So bioschemas, so what's it all about? How does it help make your resources more findable on the web? So that's what I'm hoping to get across to you today. So as humans, you may be not able to see this terribly well at the moment, but as humans, this is a web page. We can read and interpret this page. So this page is actually from the Guide to Pharmacology, and it's a list of ligands, and there's also proteins further down this page that are known to interact with COVID-19. So we can interpret that and understand and make some decisions about this page. But to machines, there's nothing on this page that helps them. Okay, it's just a string of text and they can't interpret that. That is until you add markup within it. So embedded within this page is what is called Bioschemas Markup. And this is something that makes it possible for a machine to read and interpret the contents of this web page so that it knows that there is a protein in there called ACE2, and that there's an associated disease with ACE2 that's called COVID-19. So that makes this page interpretable to the machine. Once it's interpretable to the machine, so I'm going to represent that with the Bioschemas logo, then we can do really interesting things with that content. So in the first case, that content can become more discoverable on the web through the major search engines. Secondly, we can exploit that within our informatics communities, particularly within ELIXIR. This can be used to fed into data aggregators such as identifiers.org or the Intrinsically Disordered Protein Center community, or FADAIR or OrphaNet, or the COVID-19 data portal that the EBI have set up. We can also use it to feed into larger knowledge graphs. And then this becomes part of a wider collection of knowledge about bioinformatics and can be exploited more and more widely. So how do we get started with this? So the first thing is to consider, what is the user trying to achieve? So the user is trying to find data about something. So here, I've typed in, I'm looking for data about molecular interactions. So I go to Google as a search engine, I type in molecular interactions, and I get some pages back that talk about what a molecular interaction is. It's not actually giving me any data back at this point. Okay, so I refine my search, I add in the term data to that, and I'm still not satisfied with my search result, because what I'm getting back here are papers or events about molecular interactions. I'm not actually getting the data sets themselves. So I think, okay, I've heard vaguely about this new service by Google, new being a couple of years ago now that they released it, 2018. And this is the dataset search. Okay, so let's search for molecular interactions in the dataset search. I'm looking for data. And here I get MINT back, which is great because that's a molecular interaction database. So how did this data search work? How did it know that MINT was a database of molecular interaction data and therefore a good result? Well, that's based on the markup in the MINT database pages that says this is a data set, and then that was crawled by Google and provided into their search results, particularly for their dataset search. So what markup was that using? So that markup was using something called schema.org. Schema.org was developed for web search optimization. So it's very sort of targeted at products that you might buy and sell, events, documents. It provides a structured description of the data, and it provides about 800 types. So these are the things that we can talk about. And it's got about 1,300 properties. So these are the things that we can say about those types. So in the dataset scenario, there is a dataset type. And within that, there are various properties that help me to describe my dataset. So in fact, there are about 120 properties on dataset. So how do I know which ones of those to use? Because if you look through them, you'll find that some are not so useful for datasets than others, because dataset inherits from creative work, and creative work is a very generic type. So creative work has the property is family friendly, and that's targeted at books and films, and are these family friendly, not so relevant for a dataset. So what we need then is a usage profile over the schema.org types so that we can describe the usage of this within our community. So this is where Bioschemas comes to the fore. So Bioschemas aims to do two things principally. One, it aims to extend schema.org with the types that we need to describe for things in the life sciences. So these are things like genes, proteins, diseases, molecular entity, and so on. And then once we've got these types available to us, we need to know how to make use of them. And to be honest, we're all lazy. So I'm certainly not going to mark up my data sets with 120 properties. I've got better things to do with my time. But I'm willing to put in 5 or 6 properties into my website. So I want to make sure that those are going to be the 5 or 6 that give me the most benefit for having that markup in the site. So that's where the Bioschemas profiles come in. So these are restrictions or guidelines on how to use schema.org types and Bioschemas extensions of those so that you can provide the most pertinent properties into your web pages. So these profiles have been developed by the Bioschemas community. The Bioschemas community has been going for about 6 years now, and we've engaged with at least over 20 countries, engaged with over 400 people. And in this time, we've been developing types and profiles. So we have 22 types that we've developed, 32 profiles, because some of these profiles go over types that already exist in schema.org. And we have markup on over 100 websites and over 25 million pages that we know about. So this includes markup of some of the core data resources. So Cath, EGA, HPA, Mint, Chembl, Ensemble are all in there. And also some of the data deposition databases like Biosamples, PDB Europe, and EGA. Okay, there's also many node resources that have got their markup in there as well. So I want to focus on the bioinformatics side of things here rather than schema.org side. So we've been developing new types to help describe bioinformatics resources. So we have a set of 8 released types that we've developed, and these allow us to describe things like gene, protein, and molecular entity. And what you see on the right-hand side here are some logos from the sites that have actually deployed these life sciences types. Now, in developing these types, you might ask the question, well, there's lots of bioinformatics domain ontologies out there, and you can look up these in things like BioPortal or Ontology Lookup Service, and you can find there's sort of 800 plus ontologies out there. Those ontologies are great at what they do. They model the domain semantics accurately. They model the underlying biology. What they're not so good at is helping us to discover the resources and to provide lightweight semantics by which we can use for search. And this is something that we spent some time looking at in Bioschemas in the early years. And we ended up down these rabbit holes of looking at the nitty-gritty of the biology. And it wasn't until we took this step back and realized, we're doing this for search. We're doing this to make our resources more discoverable on the web. So what are the actual pertinent properties and types that we need to do that? So Bioschemas is much more lightweight than a lot of the domain ontologies, but we have relationships to them. So we know what properties are taken from which existing domain ontology. So we haven't totally ignored them. We've used the best practice there and inherited what we can. So where does Bioschemas fit in the fair picture? Well, we're very much focused on findability of your resources. To do this, we provide globally unique identifiers. So that's your web page address. We have a community-defined enriched metadata. So that is the Bioschemas types and profiles. And ultimately, this makes your data indexable by the search engines. We also have something to say about the other parts of FAIR, so accessible, interoperable, and reusable. So your content is on the web, it's native in your web format, it's embedded as JSON, JSON-LD generally. It's using controlled vocabulary, so we link out to these domain ontologies, so we're reusing, getting that interoperability there. And we should also recommend the uses of license and provenance information to help with the reusability of the data. So how can I exploit? How do people make use of Bioschemas markup? So one great example of this is TeSS. So TeSS is the search engine for ELIXIR, about training events and courses. And this aggregates information from a wide variety of sources, at least 30 to 40 of which are using bioschemas to get their data content into the test dataset. And once it's in there, so it's using the course instance, and the training material profiles. But once that content is in there, it allows to do faceted search within the TeSS environment. And this allows you to find your resources, the training events that you're interested in a far better way. We've also been looking at connecting the content of TeSS with the content of bio.tools. So at the Biohackathon 2019, Alban was looking at how can I find which tools are associated with which training events, training materials in TeSS. And so he scraped the content from both sites, integrated that information together, and you can now have a list of, for each training event that was available at that time, what bio.tools tool is used. Earlier this year, there was the launch of the Workflow Hub, and this is all using bioschemas behind the scenes. So you can describe your workflow as a bioschemas workflow, and then use that to populate your record within the within the Workflows Hub, and then this makes your workflow more discoverable. So that's an aggregation result. We can also make use of bioschemas to exchange data without needing to go through a data repository's REST API. So MAREF was a prototype example that we used for this. So we extended the MAREF HTML with JSON. We then scraped that content and then used that to ingest into biosamples, which then meant that we could have more enriched results within biosamples with the provenance back to the original sources. So bioschemas markup can be exploited in many ways. So it can be exploited by the search engines in providing rich snippet results. It can be exploited by the search engines to provide directed portals, so with the dataset search, as that's in the top right-hand corner. It can be exploited within our own community to help power things like the TeSS the TeSS training portal search engine. And it can also be used to feed into knowledge graphs. Bioschemas has also been recommended by the International Society for Biocuration as the mechanism to provide your curations. So what tooling have we provided to help you get through and mark up your things and exploit the markup that is out there? So we've developed a Bioschemas markup generator. The generator is a simple web form. It helps you to develop the markup for your site. So you decide what sort of markup you're going to develop. You then fill in the questions in the form, so fill in the blanks, and then hit the generate button at the bottom of the form. And then that will output the JSON LD that you can adapt or just place straight into your website. Once you've placed your markup within your website, you're going to want to check that it's retrievable and that it conforms with the profile. So we've been developing a validator for that. So the validator makes use of the crawler, or the scraper that I'm going to describe next, and pulls in that live code, although you can also paste in sample code as well. And then you choose the profile against which you want to check your markup against and hit the validate button. And then you get at the bottom either a green light or a red light. If you get a red light, I'm afraid our error reporting isn't great at this point, but that's something that we can move on and innovate on in the future. Finally, we have a scraper. So a scraper and extractor to be able to pull back the markup that has been embedded in websites. So this can deal with both single page applications and static sites. It also returns the provenance information of where and when that information has been scraped. And it can also pull back that content regardless of whether it's been embedded as JSON LD or RDFA. So you can seed that with a set of URLs or a set of sitemaps or a combination of both. And this will do a directed scrape of that content. So this is going to be useful for developing community aggregators of information. So one example that we've been trying out at Herriot-Watt is a targeted scrape of COVID information. So we've started with this with some sites that we knew had COVID markup within them, or markup about their COVID available resources, and we've been starting to scrape that content. So that's been making use of our Bermudes. Within ELIXIR, we are targeting working with lots of different communities. So I'm going to mention a few here, although there are a wider set of communities that we are also discussing with. So at the moment, we have an implementation study that's working within the rare disease community. So we're looking here to develop aggregation of the long tail of information that's available about rare diseases, because there's a lot of smattering of information out there on the web, but not very many large repositories of that. So what we want to do is use disease profiles and use that to feed into a repository such as OrphaNet to make as much information about these rare diseases available as possible. Similarly, we need to know about the genes that affect those rare diseases. So we're going to be pulling in that gene information. So this will make use of that directed scraper that I was just talking about. Within the plant community, well, the plant community already have findability as a search portal, and this uses information that is harvested using breeding API endpoints. But to set up a breeding API endpoint, you need to have a lot of infrastructure. You're setting up a web service with a REST API, and that's not necessarily possible for all people. So we also want to augment that information with the content of smaller sites who are maybe providing gene information, phenotype information, and have that available through the findability search portal as well. So making the best of both worlds. And then within the intrinsically disordered protein community, they are at the early stage of developing a DisProt Central. They have a lot of data sets that have information about intrinsically disordered proteins. These are gradually getting the protein profile and protein structure markup within them. And again, we're going to set up this directed crawl to be able to have a central data aggregation for the community that makes this information available. And the similar thing is going on in the toxicology community. Again, we've got a series of data sets, a central aggregator, and we're trying to make this information more available from a central place. So I've shown you what is Bioschemas markup. I've shown you how it's being exploited and its potential. So what I want to talk about just briefly at the end here is how you might develop that markup. And where markup actually exists. So as I said at the start, we have over 100 sites with markup deployed on them. So you can find these on our live deploys page. I've taken a few screenshots from this with the molecular entity expanded. So here you can see that there are 4 resources that have molecular entity markup within them. And if we look into one of these in more details, we'll look at MassBank. And again, here I've got a human readable page. Okay, so the markup doesn't affect that human experience of the web. We still have native HTML pages there. But within this page, there is markup embedded. And this is embedded as a chunk of JSON-LD in this case. And what I've used here is the Google Structured Data Testing tool to show what this markup looks like. Okay, so it's just a block at the end of the page of JSON-LD. And then this can be extracted to see that this has information about something that is a molecular entity. It's got an identifier, it's got URL, a name associated with it, and a molecular formula. The choice of these properties has come from the Bioschemas profile. So the profile here is the molecular entity profile. Each profile has a set of minimal properties that we feel that people absolutely must provide. These are generally so that we've got something to return to the user. And then there's a set of recommended properties. So these are properties that are not always absolutely necessary to be there, but it's good practice to put them there. And these are useful for discriminating between search results. So in the case of molecular entity, we want to know what the identifier, so the URI of the page is, and the name of the molecular entity. And then we recommend that people provide the INCHI, the INCHI key, the IUPAC name, and so forth. As I said before, to help you do that markup, we have some tooling to help you. So the steps that you would go through, the recipe to help embed that markup in your page is, first of all, decide on the type of markup that you have. So what is your page about? So here I have the page from the Natural History Museum in Paris, and this is a page about a taxon. So they would choose the taxon profile to generate their markup with, and then they would look at the properties that they have on this page and look at the profile and connect the two together. So we've got a name here, we've got a description, so these would be used to link to the profile properties. Alternatively, you can use the Bioschemas markup generator. Sorry, I jumped over that. And you can just fill these into the blanks in the generator and then hit the generate button at the end. And as I say, that gives you a block of JSON LD back, and you just then embed that JSON LD within your website template. So you can replace the actual value of the name with a database parameter, so that when you put this in your templating engine in your website, it just populates that field with the value there. So here you can see that the script is an application JSON LD. It's got a context of schema.org, and then the properties appear. So just as a summary, so Bioschemas, it's about making your web pages about your resources more discoverable on the web. We do this by embedding Bioschemas markup within that. Once you've got your markup in there, it can be scraped, it can be crawled by anyone and everyone, and that makes your data more reusable. But we can also ensure that people provide those links back because we're providing that provenance information based on the scraping that we're doing. So the major search engines can make use of it. Our community, the ELIXIR community, can make use of your data, and it can also go out into the wider scientific community. So Bioschemas, as I said, is a big community of individuals. This is just some of the people who've been more involved with the development of the profiles. There are multiple ways that you can connect with the community. So we have a Slack channel. You can find us on GitHub. You can find us on Twitter, and you can also find the website. If you're interested in finding out more and getting more involved with the community, we have a community call on the 4th Monday of every month. The next call is on the 26th of October at 1500 GMT. And we're also looking to expand the steering council of Bioschemas at the moment. So if you fancy getting more involved at that level, you can join the steering council. And that's all I'd like to say about Bioschemas today.